Hello guys, I welcome you all to Unacademy on the CSIR NET platform, and we are solving the important MCQ series. Myself Himanshu. Let's begin today's session. Good afternoon everyone. तो स्टार्ट करने से पहले मैं आपको बताना चाहूँगा कि you need to go to the Google Play Store where you need to install the Unacademy Learning app, and after you install the Unacademy Learning app you need to select your goal in you need to do login. So once you do login, you'll be getting CSIR UGC NET category, in which you will be getting access to various free courses from various educators. If you want to get exclusive guidance, then you need to take the plus subscription, where you'll be getting mock test, you'll be getting quizzes, you will be getting notes. So for getting the plus subscription, you will be required to make subscription for CSIR UGC NET, and the subscriptions are available for 1 month, 3 months, 6 months, 12 months and 24 months. So I would recommend that you go for the subscription of 12 months to 24 months. You can always use my code HY10, where you will be getting 10% off. So after applying my code HY10, you will be getting 10% off. You need to proceed to the payment, where you will be getting the credit card and the debit card. So you need to enter the card number. You need to make the payment. You can also do the payment by the EMI, and these EMI are no cost EMI. You can also do the payment by net banking. So this is all. So let's begin today's session. तो ये पहला क्वेश्चन है. So in an experiment, intact chromatin was isolated and digested with micrococcal nucleus in independent tubes for 30 minutes, 1 hour, 2 hour and 4 hour. Further, the DNA was purified from each tube and separated on agarose gel, and southern hybridization was performed with rRNA gene probe and centromeric DNA probe. Which one of the following patterns of signal intensity from both of the probes is likely to be obtained following southern hybridization? So option A says with increasing time compared to centromeric probe, a rapid increase in signal intensity of the rRNA gene probe was observed. Option B says with increasing time compared to the centromeric probe, a rapid decrease in the signal intensity of the rRNA gene probe was observed. Option C says irrespective of the incubation period, both the probes produced identical band and intensities. And option D says treatment with micrococcal nucleus would instantly degrade the DNA. Hence, no hybridization signal would be obtained in any of the samples. तो इस क्वेश्चन को आप लोग अच्छे से पढ़िए. So in an experiment, the intact chromatin was isolated and digested with micrococcal nucleus in independent tube for 30 minutes, 1 hour, 2 hours, 2 hours and 4 hours. Further, the DNA was purified from each tube, separated on agarose gel, and southern hybridization was performed with rRNA gene probe and centromeric DNA probe. Which one of the following patterns of the signal intensity from both the probes is likely to be obtained following southern hybridization? So with increasing time compared to centromeric probe, इसमें पूछ रहा है which one of the following patterns of signal intensity from both the probes is likely to be obtained following southern hybridization. So with increasing time compared to centromeric probe, a rapid increase in the signal intensity of the rRNA gene probe was observed. So this is a wrong statement. With increasing time compared to the centromeric probe, a rapid decrease in the signal intensity of rRNA gene probe was observed. This is the correct option. Irrespective of the incubation period, both the probe produce identical band intensity. So both the probe produce identical band intensity. So this is a wrong option. Treatment with micrococcal nucleus would instantly degrade the DNA. Hence, no hybridization signal would be obtained in any of the samples. So option B is the correct option. जैसे जैसे टाइम बढ़ेगा compared to the centromeric probe, a rapid decrease in the signal intensity of the rRNA gene probe was observed. So option B is the correct option. यहाँ पे ये बताया गया कि माइक्रोकोकल न्यूक्लियस एक एंडोन्यूक्लियस है, वो क्लीव करता है बोथ सिंगल स्टैंडर्ड एंड डबल स्टैंडर्ड डीएनए एंड आरएनए. It yields oligonucleotides to mononucleotides depending on the incubation time of the DNA in the micrococcal nucleus solution. That is, longer the duration of incubation with micrococcal nucleus, shorter will be the fragment. So the length of centromeric DNA in humans is around 0.2 to 7 megabases, while rRNA genes are in the range of 120 to 2000 base pairs. Thus, with the longer duration of the nucleus, one will find decreasing intensity for shorter length of DNA. To digest centromeric DNA, more incubation time will be needed. Therefore, with time, the degradation of rRNA will be conspicuous on southern blot upon probing. So this is the explanation. So let's move to the next question. A single protofilament of microtubule grows at a speed of 2 micrometer per minute. Considering that there is no catastrophe in the microtubule nucleation and the size of tubulin unit is of the order 5 nanometer, how many tubulin units are added to the growing microtubule per minute? So option A says 400, option B says 1600, option C says 3200, and this is option D which says 5200. So इसमें option D is the correct option. So since the protofilament is growing at a speed of 2 micrometer per minute, that is 2000 nanometer per minute. The unit of protofilament is 5 nanometer in size. Therefore, number of tubulin units added to the growing protofilament will be 2000 by 5, that is 400 tubulin units per minute. A microtubule is itself made of 13 protofilaments. So if a single protofilament is growing by 400 tubulin units, then 13 filaments will have 400 into 13 tubulin units. Hence, correct option is D, as 5200 units were added to the growing microtubule per minute. So this is the explanation. So let's move to the next question. This one is the D option. An intron in the yeast reported gene carries a mutation in the splice site branch point. To suppress the mutation, a library of point mutants of snRNA was introduced into the mutant strain. The suppressor is most likely to have a mutation in this one is the UN snRNA. This is option B is U2 snRNA. Option C is RNase polymerase. Option D is U6 snRNA. सो करेक्ट ऑप्शन को आप लोग टाइप करिए कमेंट बॉक्स में. So U2 snRNA is the correct option. Let's move to the next point. So option B is the correct option, as we have seen. So highly conservative splice site sequence UACU 3 prime is 10 to 50 base pairs upstream from the 3 prime site of the intron in yeast. This sequence is crucial for U2 snRNA to recognize for accurate splicing. Therefore, if the sequence site of splicing is mutated in the reported gene, then the recognition site of U2 snRNA must also be mutated to suppress the mutation. The UN snRNA interact with the 5 prime splice site of the intron for pre-mRNA splicing. U6 snRNA also is a part of spliceosome, but it is not directly involved in the branch point recognition. The processing of tRNA is done by RNA polymerase. Thus, option C is incorrect. So option B1 is the correct option. Which one of the following statements regarding clonal selection hypothesis is not correct? Mature B lymphocytes bear Ig receptors on their cell surface. and all the receptors on a single B cell have variable specificity for antigen. Okay. तो इसमें पूछ रहा है which of the following statement regarding clonal selection hypothesis is not correct. So and it is saying that all the receptors on a single B cell have variable specificity for antigen. So this is a wrong statement. On antigen stimulation, B cell matures, okay, migrates to lymphoid organs and replicates. Its clonal descendant bear the same receptor as the parental B cell and secrete antibodies with identical specificity. This one is a correct option. So after an immune response, more B cell bearing receptors will remain in the host and act as memory cells for mounting enhanced secondary response. So this is also a correct option. B cell with receptors for self antigens are deleted during embryonic development. So this is also a correct option. So option A is incorrect. So option A is the correct option. So option A is the correct option. So the clonal selection hypothesis revolves around the main idea to describe how the immune system distinguishes between self and foreign presence by B cell. This is one of the essential hypotheses in immunology, hence memorizing it will be helpful. Although the mature B cells indeed have immunoglobulin receptors, their specificity for the antigen remains the same. Therefore, option A is incorrect. Which one of the following statements on protein conformation is not true? Next question is, which one of the following statements on protein conformation is not true? Option 1 says dihedral angles of side chains in amino acids are depicted in the Ramachandran plot. Infrared spectroscopy can be used to deduce hydrogen bonding in peptides. So this is the correct option. Three-dimensional structure of protein composed of 100 amino acids can be obtained by nuclear magnetic resonance spectroscopy. Okay. So this is also a correct option. Globular proteins have alpha helical and beta sheet components. So this is also a correct option. Dihedral angles of side chains in amino acids are depicted in the Ramachandran plot. So this is a wrong option. This one is not true. So statement 1 is not true. So Ramachandran plot describes the stable phi and psi angles formed by the main chain of amino acids and not the side chain. Therefore, option A is incorrect. Two-dimensional infrared spectroscopy exchanges hydrogen with deuterium, and consequently the dynamics of hydrogen bonding can be studied. So option B is correct. The 3D or native structure of protein can be determined by NMR as it measures the distance between the nuclei of the atoms around it, which further is useful in determining the structure of the molecule. Hence option C is correct. Globular proteins are the approximately spear-shaped proteins which are also abundant in nature. So they have abundant alpha and beta folds in their structure. Therefore option D is also correct. So let's move to the next question. Next question says that an autosomal recessive condition affects 1 in 10,000 in a random mating population without any disruptive acting force. What is the approximate expected frequency of carriers in the population, in this population? So option A says 1 in 10,000 newborns, option B says 1 in 500 newborns, option C says 1 in 100 newborns, and option D says 1 in 50 newborns. So option D is the correct option in my opinion. Let's see the correct option. Yes, option D was the correct option. Since no external disruptive force is acting and the population is mating at random, the stated community is at Hardy-Weinberg population. The population will follow capital P plus small p equals to 1 and capital P square plus 2p 2 capital P small p plus p square, where capital P and small p are alleles. So the recessive genotype P square causing disease occurs in 1 in 10,000. Thus the genotypic frequency will be 1 upon 10,000, that is 0.0001. The allelic frequency, that is small p, is 0.01. Therefore the allelic frequency of capital P will be p plus 0.01 equal to 1, that is 0.99. So carriers will be heterozygotes and thus their probability will be 2 into 0.01 into 0.99, that is 0.0198. So overall they will comprise of 198 individuals in 10,000 people. So by unitary method, if 198 carriers are found in 10,000, then one carrier will be found among 50.50, which is approximately 50, making option D correct. तो इस एक्सप्लेनेशन को आप अच्छे से देख लीजिए. Let's move to the next question. Complete the following sentence with the most appropriate option. Global analysis of large number of plant species traits showed that with an increase in the leaf lifespan, option A says specific leaf area increases whereas leaf nitrogen and net photosynthetic rate decreases. Option B says specific leaf area, leaf nitrogen and net photosynthetic rate increase. Option C says specific leaf area, leaf nitrogen and net photosynthetic rate decreases. Option D says specific leaf area decreases whereas leaf nitrogen and net photosynthetic rate increases. तो इस क्वेश्चन को अच्छे से पढ़िए और करेक्ट ऑप्शन को टाइप करिए कमेंट बॉक्स में. तो यहाँ पे बता रहा है कि लार्ज नंबर ऑफ प्लांट स्पीशी ट्रेड शो दैट विद एन इंक्रीज इन लीफ लाइफ स्पैन specific leaf area increases whereas leaf nitrogen and net photosynthetic rate decreases. So this is a wrong option. Specific leaf area and leaf nitrogen and net photosynthetic rate increases. So this is again a wrong option. Specific leaf area, leaf nitrogen and net photosynthetic rate decreases. So this is the correct option. And specific leaf area decrease whereas leaf nitrogen and net photosynthetic rate increase. This is again a wrong option. So option C is the correct option. So studies have shown that the plant resources will be utilized either for the growth or for photosynthetic assimilation, but never both. Therefore, if the leaf lifespan is increased, then the specific leaf area, leaf nitrogen and net photosynthetic rate will decrease as the energy will be consumed for growth. So this is the correct option. So plant resources will be utilized either for the growth, that is the increased leaf lifespan, or for the photosynthetic assimilation, but never both. Therefore, if the leaf lifespan is increased, then the specific leaf area and leaf nitrogen and the net photosynthetic rate will decrease as the energy will be consumed for growth. So let's move to the next question. This one is the D option. So in order to estimate the population size of the fish species in lake, a researcher captures 100 fish from the lake and marks them with color tag. A week later, the researcher returns to the lake and catches 150 fishes of the same species and finds that 25 of them are previously tagged ones. Assuming no immigration or emigration have occurred, the total population size of the fish species in the lake will be. Option A says 17, option B says 38, option C says 600, option D says 860. तो आप लोग अपने ऑप्शन को कमेंट बॉक्स में टाइप करें. So let's move to the solution. So this is a capture recapture. It is one of the widely used method to estimate the population of a species in their natural habitat. So the general formula for this method is capital N upon small m, capital N upon capital M is equals to small n upon small m, where capital N are the estimated total population, where n is equals to n. M is the number of fish captured in the first attempt, so m will be 100. Small n is the total number of fish caught in the second attempt, so small n will be 150. And small m will be total fishes that were already tagged, that is 25. So by putting the options in the values, so we have to calculate capital N, so which is the estimated total population. So n by 100 will be equal to 150 by 25. So n will be 150 into 100 upon 25. So this one will be equal to 600. So option C is right. Given that no immigration and emigration have occurred, so option C is the correct option. This one is based on the mark and recaptured method. So thank you guys. If you have any doubts, you can post it in the comment box. अगर आप लोग के कोई भी डाउट्स या क्वेश्चंस हैं तो आप लोग कमेंट बॉक्स में पोस्ट कर सकते हैं. And please like and share and subscribe to this channel. Like and share as much as possible and also subscribe. And please share with all those who are preparing for this exam. And you can use my referral code HY10 for getting 10% off on next Unacademy subscription. So this is my referral code. So thank you guys.